Chemistry of protein. Chapter 3. Amino acid is the amino group NH2 containing the carboxylic acid. Each amino acid has an acidic carbohydroxyl group COOH, a basic amino group NH2, and a distinct hydrocarbon side chain R group. NH2 group, COOH group, and the distinct side chain R are bound to the A carbon. General structure of amino acid: CH3, CH2, and CH, So amino acids have amino acid has a central asymmetric A carbon to which following four different amino acid groups are attached. Amino group NH2 is isoelectric, pH is 9, so that body pH 7.4. It exists as protonated from NH3 plus. Carboxylic group COOH is isoelectric, pH is 2, so at body pH 7.4 it exists as deprotonated from COO minus. Hydrogen atom H and side chain R.
Is the group of seed Iterbiceae differ from each other only with respect to the side chain. So the nature of the side chain determines their properties and function in protein. In total, there are 20 different amino acids in human body, and all body proteins are constructed from this basic set of 20 amino acids, which are specified by the genetic code. So they are also called the primary amino acid or standard amino acid. Amino acid participating in body protein synthesis are L-alpha type because the amino group is usually attached with the alpha carbon and placed on the left side with respect to the special configurations. Alpha carbon with NH2 group or left side chain. All body amino acids except glycine have the asymmetric alpha carbon, so amino acid can produce optical isomers with optical activity. Exceptionally, the glycine does not have any side chain R. Here R is replaced by another hydrogen atom. Therefore, its alpha carbon is systematic. Conventionally, name of amino acids are abbreviated by the first three letters of their name. Example, the serine. Exception to this convention is practiced only for the tryptophan, isoleucine, asparagine, and glutamine. The classification of amino acids based on a structure of side chains and their reaction in solution. Acid amino acid, monoamino dicarboxyl acid, negatively charged at body pH. They contain one amino group and two carboxyl groups. Aspartic acid and glutamic acid. Basic amino acid, diamino monocarboxyl acid, positively charged at body pH. They contain two amino groups and one carboxyl groups, lysine, arginine, and histidine. The natural amino acids carry no net chains at the body pH. They contain one amino group, one carboxyl group. There are three types: aliphatic amino acid, aromatic amino acid, amino acid. Aliphatic amino acid, sulfur containing cysteine and methionine, and branch chain valine, leucine, isoleucine. Hydroxyl group containing serine, threonine. Amide group containing asparagine and the glutamine. Simple structure: glycine and alanine. Aromatic amino group: phenylalanine, tyrosine, and tryptophan. The amino acid proline, it contains amino group and not NH2 group. Based on polarity of the side chain, polar amino acid, they contain hydrophilic side chain. Acidic amino acid, basic amino acid, glycine, serine, threonine, cysteine, glycine, acid. Non-polar amino acid, they contain hydrophobic side chain. Mat, vel, liu, ala, tryptophan. The nature classification of amino acid. Essential amino acid, indispensable amino acid. Amino acid which are not produced in body and must be supplied in diet. Their presence in diet is essential, so they are called essential. Essential amino acids are derived mainly from the animal protein, meat, fish, egg, milk. Example: the phi, bay, trip, tree, ili, mat, liu, The pill is useful for the recalling the names of essential amino acids amid tryptophan and tree. Pvel, lu, lysis. Semi-essential amino acids. They are produced in body but in limited rate, because of which they cannot meet up the body demand when their requirement is high, and during growth in infants and children, but they can meet up the body demand when their requirement is less in adult when there is no growth. Therefore, they are essential for infants and children, but no essential for adults. So they are called the semi-essential, arginine and histine. No non-essential amino acid or dispensable amino acid. These are produced in body in adequate amount, so their presence in diet is not essential, so they are not essential amino acid. Aspirin, glue, crystal, ser, gly, ella, acin, gly, tree, pro. Metabolic calcification. Metabolic calcification of amino acid based on their catabolic fate. Absolute glucogenic amino acid. These are the amino acid having carbon skeleton that process the metabolic potential to produce glucose. In fact, they initially produce precursor of the glucose. Example: pyruvate and intermediate from which later on the glucose is produced. The glycine, ella, ser, gris, pro, glu, glyne, aspirin, acetylgen, arginine, histine, mat, pre, well. Absolute ketogenic amino acid. These are the amino acids having carbon skeleton that process metabolic potential to produce the ketone body. In fact, they initially produce precursor of ketone bodies, acetyl-CoA or acetyl-CoA, from which later on the ketone body is produced. Leucine, lysine, both glucogenic and ketogenic amino acid. There are the amino acids having carbon skeleton that process metabolic potential to produce both glucose and ketone body. In fact, they initially produced acetyl-CoA and acetyl-CoA along with pyruvate or TCA cycle intermediates, and then produce the ketone body or glucose. Phenylalanine, triosine, and tryptophan and isoleucine. Nutritional and metabolic classification of amino acid. Metabolic type. Nutritional type: essential, non-essential. Absolute glucogenic: histidine, metrin, 3-valine arginine. Ella, aspirin, acin, glucoglycine, pristine, cereal, glycan, protein. Both glucogenic and ketogenic. Phyllate, trip, iodide, tray. Absolute ketogenic, leucine. Properties of amino acids. Properties of amino acid: physical properties, chemical properties. Colorless, crystalline, usually water soluble. Process high melting point, more than 200 degrees centigrade. Chemical properties: act as ampholytic, behave both an acid and base at body pH, and also act as buffer. Carboxyl group act as acid group, acetic group by donating protein, and amino group act as basic group by accepting protein. Express charge on the surface and have the definite isoelectric pH. Isoelectric pH of amino acid is the pH at which amino acid exit as Jupiter ion, possessing same amount of positive and negative charge on the surface with net charge 0. At isoelectric pH, amino acid shows no electrophoretic mobility, become least soluble, and rapidly participate out of solution. Depending on the isoelectric pH of amino acid and the pH of surrounding media, each amino acid can exist in either of the three charged state, anion, cation, and natural Jupiter ion.
Undergo carboxylation to form corresponding amines. amines. Form ester with alcohol and form salts with acid or base. Give purple color reaction with ninhydrin and form carbono compound with CO2. Functions of amino acid act as the building blocks of peptide, polypeptide and protein. Support gluconeogenesis in fasting and starvation. Functions of amino acid synthesis of socialized products, for example neurotransmitters, hormones, purine, pyrimidine, heme and glutathione, etc. Functions of amino acid acts as the building blocks of peptide, polypeptide and protein. Support gluconeogenesis in fasting and starvation. Detoxification of toxic substances in liver by glycine, cysteine, etc. Act as metabolic fuel to provide energy. Meet up 10% of the body energy needed. Arbitrarily peptide, polypeptide and protein and biomolecule made of 2 to 10 amino acids, 10 to 100 amino acids and more than 100 amino acids respectively connected by peptide bonds. Some important peptides: thyropropine releasing hormone TRH, number of amino acids 3. Glutathione, oxytocin 9, ADH 9, bradykinin 9, number of amino acids. Angiotensin 1 10, angiotensin 2 8. Definition of protein: macromolecular polymer of amino acid linkage together by peptide bonds. Usually a protein consists of more than 100 amino acids with minimum molecular weight 5000 to 8000. Classifications of protein based on size, shape and solubility. A simple protein composed of amino acid residues without any non-protein substance and non-hydrolysis. They produce only amino acids. There are two types: fibrous protein, scleroprotein, for example collagen, elastin and keratin, entirely animal origin and have high molecular weight. Fiber-like with high tensile strength, water insoluble and highly resistant to proteolytic enzyme. Found in tendon, ligament, cartilage, extracellular matrix, hair, nail, etc. Globular protein, spheroprotein, for example albumin, globulin, protamine, histone. Oval or spherical in shape with relatively low molecular weight, water soluble and digestible by proteolytic enzyme. Conjugated protein, composed of protein along with non-protein protetic substance. On hydrolysis they produce amino acid plus other non-amino acid substance. Conjugated protein, prosthetic group, nucleoprotein, nucleic acid, lipoprotein, lipid, glycoprotein, carbohydrate, ferritin, iron, carbonic anhydrase, zinc, ceruloplasmin, copper, hemoglobin, myoglobin, cytochrome, peroxidase, catalase, rhodopsin, retinol, ferritin, carbonic anhydrase and ceruloplasmin are called metalloprotein. Heme and retinoprosthetic group containing protein are called chromoprotein. Hemoprosthetic group containing protein are also called hemoprotein. Derived protein, denatured or degraded products of simple protein and conjugated protein, for example proteose, peptone, polypeptide and peptides. Functional classification of protein: types of protein, function and example. Structural protein support framework of cell, collagen and elastin, etc. Catalytic protein, catalysis enzymes. Transport protein, transport of substance, albumin transferrin. Hormonal protein, regular functions, insulin, glucose, etc. Gene regular protein, regulate genetic function. Histone protamine, protective protein, prevent infection, immunoglobulin. Respector protein, respector, LDL respector. Contractile protein, muscle contraction, actin, myosin. Nutritional classification of protein. Complete protein, they contain all essential amino acids and promote growth. For example, egg, albumin, meat, etc. Incomplete protein, they completely lack one or more essential amino acids and cannot promote growth. For example, gelatin, it lacks TRP. Partially incomplete protein, they partially lack one or more essential amino acid. For example, wheat protein. Elementary composition of protein: carbon 55%, oxygen 25%, hydrogen 7%, nitrogen 20%, average 16%, sulfur 4%, nitrogen 16%, carbon 55%. Peptide bond formation: it is a covalent bond that connects adjacent amino acid to form peptide, polypeptide, and protein. It is formed by joining the COOH group of one amino acid and the NH2 group of another amino acid with removal of one molecule water. Amino and or N terminal, peptide bond, peptide unit, C terminal. Pi, converting free NH2 group of amino acid donating COOH group to peptide bond. It is written to the left and the free COOH group of amino acid. Characteristics of peptide bond: this is a rigid planar covalent bond and shows particle double bond character, not broken by denaturing agents but broken by proteolytic enzyme. Okay. Structure of protein usually described at 4 levels of organization. 1. Primary structure. Primary structure, it is the exact linear sequence of amino acid in protein held together by peptide bonds that determines the type, quantity, and sequence of amino acid in protein. Each amino acid is a polypeptide chain is called amino acid residue because it is the remaining protein of amino acid after the atoms of water has been lost during peptide bond formation. The free NH2 group of one terminal amino acid. The free NH2 group is one terminal amino acid is called amino and terminal, and the free COOH group is another terminal acid is called carboxylic and C terminal. Traditionally, amino and is assumed to be one left hand side with carboxyl and on the right side. By convention, the sequence of amino acid is written from right to left, starting from the amino acid and amino end as number 1 and then proceeded towards the carboxylic end. Presence of a specific amino acid at a specific position is very significant from a particular function of protein, and any changes in the sequence is abnormal, which may affect the properties and functions of protein. For example, sickle cell anemia occurs due to replacement of normally present glutamate at 6th position of alpha chain of hemoglobin by valine. Primary structure of a protein is determined genetically, and it is determines secondarily tertiary and quaternary structures. First amino acid, secondary structures. Tertiary structure. Secondary structure, it is a helical or pitted sheet like information of protein produced by periodic folding, twisting, or coiling of the primary structure. tertiary structure. It is the tertiary structure. It is three-dimensional globular form of protein produced by further folding the and twisting of secondary structure about itself with the hydrophobic side chains buried interiorly and hydrophilic groups exposed outside, also found in albumin, globulin, etc. Quaternary structure. Quaternary structure. It is multi-chain oligomeric protein, shows special relationship of individual polypeptide chain among each other. Protein composed of a single polypeptide chain is called monomeric protein, and that of two or more polypeptide chain is called oligomeric protein. Properties of protein. A physical properties. Macromolecule with variable shape and variable molecular weight. Chemical properties. Amphoteric in nature and can act as a buffer. Therefore, the electric pH of protein is used to separate proteins by chemical methods or by electrophoresis. Page 31-32 gives color creation with ninhydrin, burette, reagent, etc. Biological importance. Function of protein. Formation of skeleton. Provides cytoskeleton is a complex network of protein filaments in cytoplasm that forms a flexible structure framework for cells and tissues. Provide chemical support by structural protein, for example, collagen, elastin. Promote catalytic function by enzyme and hormonal function by hormones. Provides defense against the infection by antibody. Immunoglobulin helps in muscle contraction by contract protein. For example, actin, myosin acts as vehicle for transport and hormones, drugs, vitamins, trace elements, etc. Plasma protein containing colloidal osmotic pressure of plasma to maintain blood volume. Helps in coagulation by clotting factors and act as buffer. Storage, expression and transmission of genetic information by nucleoprotein, source of energy. Denoted protein. Proteins produced following loss of their native structure conformation due to disruption of secondary, tertiary and quaternary structure but preserving the primary structure. Coagulated protein, i.e. boiled egg, cooked meat, cooked fish. Proteins produced by irreversible denutrition with intramolecular change. They are insoluble and precipitate out. All coagulated proteins are denoted protein, but all denoted proteins are not coagulated protein. All denoted proteins are denatured proteins are not coagulated proteins. Fibrous protein, collagen. It is the most abundant protein in human body. 25% of total protein found in extracellular matrix, skin, bone, cartilage, ligament, cornea, etc. Collagen. Elastin. Elastin is a fibrous protein with rubber-like properties, elasticity that can be stretched and again recoil. Globular protein, plasma protein. Functions of albumin plasma protein. In case of hyperalbuminemia, plasma COPD decrease and intravascular fluid does go out of blood vessel into the interstitial space to develop edema. Functions of globulin. Vitamin D. In important globulins transported substances. Vitamin D binding protein DBP. Vitamin D retinobinding protein retinol. Transcobalamin. Vitamin B12 thyroxine binding protein. Thyroid hormones. Sex hormone binding globulin. Sex hormone, for example, testosterone, oestradiol. Cholesterol binding protein cholesterol. Haptoglobulin, hemoglobin. homotaxin heme, transferrin iron, ceruloplasmin copper. Hemoglobin. Chemistry of Hb. Hemoglobin is conjugated protein made of hemo, non-protein, and globin, protein. Globin is composed of 4 polypeptide chains. Adult hemoglobin HbA is a tetramer with 2 alpha polypeptide chains, 2 beta polypeptide chains, and 4 heme. Every polypeptide chain is conjugated with one molecule of hem. Type of hemoglobin. Hemoglobin may be different types depending on the globular peptide chain composition, but the hem part is the same in all hemoglobinates. For example, HbA type of hemoglobin HbF, HbA2, alpha 2, beta 2, alpha 2, gamma 2, alpha 2, delta 95%, less than 2%, 2 to 5%. Functions of hemoglobin. Difference between HbA and HbF. Affinity to oxygen less, more tendency to unload oxygen more, less. Synthesis of hemoglobin. Synthesis of hem. Synthesis of protoporphyrin IX from succinyl CoA and glycine. Combination of 4 subunits to make hemoglobin. Succinyl CoA plus glycine, protoporphyrin IX, iron hem, globulin, polypeptide chain, 1 subunit hemoglobin. Combination of 4 subunit. Chapter 4. Chemistry of lipid. Chemistry of lipid. Chapter 4. Definition of lipid. Lipids are heterogeneous group of organic substances which are water insoluble but soluble in fat solvent. Common fat solvents are ether, chloroform, benzene, alcohol, etc. Protein, polysaccharide, and nucleic acid are polymer, but lipids are not polymeric substances. Rather, they are mostly small molecules. Building blocks of most of the lipids are fatty acids. Classification of lipid. A simple lipid, for example, neutral fat and wax. Ester of fatty acid and alcohol. They don't contain non-lipid substance. Basically composed of fatty acid plus alcohol. B. Complex compound lipid, for example, phospholipid, glycolipid, and lipoprotein. Ester and fatty acid with alcohol along the other non-lipid substance. Basically composed of fatty acid plus alcohol plus non-lipid substance. Derived lipid. Derivatives obtained by hydrolysis of simple lipid and complex lipid which still possess the general characteristics of lipid. For example, fatty acid, alcohol, glycerol, steroid, cholesterol, prostaglandins, fat soluble vitamins, ketone bodies, etc. Neutral fat. Ester of fatty acids with glycerol. It is also called triglyceride (TG) or triacylglycerol (TAG) or oil. Chemically, they are composed of one glycerol molecule with 3 fatty acids, and they are regarded as natural because they do not have any charge at normal body pH. Fat in liquid state is called oil. For example, vegetable oil, fish oil. Fatty acids found in natural fat are mostly palmitic acid, stearic acid, and oleic acid. Neutral fat of fat source. For example, vegetable oil have more unsaturated fat acid than the of animal source. For example, ghee, egg, etc. They have more structured fatty acid. Difference between fat and oil. Fat, solid at room temperature 25 degrees Celsius, contain more saturated fatty acid. Oil, liquid at normal temperature, contain more unsaturated fatty acid. This difference is purely physical. Chemically, both are same. triacylglycerol, and made of fatty acid and glycerol. Wax, ester of fatty acid with long chain monohydric alcohol. For example, retinol ester, retinol plus fatty acid; cholesterol ester, cholesterol plus fatty acid; vitamin D ester, vitamin D plus fatty acid; phospholipid, ester of fatty acid Fa with alcohol attached to phosphoric acid with or without nitrogen base, basically composed of fatty acid plus alcohol, phosphoric acid with or without nitrogen base. Phospholipid, ester or fatty acid Fa with alcohol attached to phosphoric acid with or without nitrogen base, basically composed of fatty acid, alcohol, phosphoric acid with or without nitrogen base, but in most of cases nitrogen base is present. Nitrogen base is not essential component, but H3PO4 is essential component of phospholipid. Depending on the nature of alcohol present, phospholipids can be classified into glycerophospholipid and sphingophospholipid. Glycerophospholipid, here the alcohol is glycerol, so basically it is composed of fatty acid plus glycerol plus phosphoric acid with or without nitrogen base. Fatty acid are attached with first and second carbon, and phosphate is attached with third carbon, glycerol. Glycerophospholipid contains at least one unsaturated fatty acid, usually at second carbon, C2 or glycerol. Primary glycerophospholipid is phosphatidic acid composed of fatty acid, glycerol, and H3PO4. Others are derivatives of phosphatidic acid. Phosphatidic acid, a glycerophospholipid, a glycerophospholipid without nitrogen base. Here R represents fatty acid. Glycerophospholipid with nitrogen base, phosphatidic acid plus nitrogen base. Major fatty acids found in glycerophospholipid are polyunsaturated fatty acid, essential fatty acid, monounsaturated fatty acid, oleic acid, palmitoleic acid, saturated fatty acid, and palmitic acid. Stearic acid, commonly glycerophospholipids and their composition. Glycerophospholipid composition, nitrogen base. Phosphatidic acid, Pa, lecithin, sapalin, phosphatidylserine, phosphatidylиnositol, phosphatidylglycerol, lysophospholipid. Absent chlorine, ethanol, amine, serine, absent, mostly present. Sphingophospholipid, sphingomyelin. Here the alcohol is sphingosine. So basically it is composed of fatty acid plus sphignol, phosphoric acid plus choline. Fatty acid and sphignol together make ceramide. So alternatively it can be stated that sphingomyelin equal to ceramide plus H3PO4 plus choline. Unlike other phospholipids, they contain mostly 16 to 26 carbon unsaturated fatty acid, for example palmitic acid, stearic acid, etc. Here H3PO4 and choline together represent non-lipid substance, and choline itself represents nitrogen substance. Sphingol fatty acid, H3PO4, choline. Sphingomyelin constitute biological membrane and myelin sheath. Glycolipid, sphingolipid, they are a lipid containing carbohydrate, basically composed of fatty acid plus sphingol. carbohydrate or ceramide or carbohydrate. Here fatty acid with sphygmo represents lipid part and carbohydrate represents non-lipid part. Carbohydrates present in glycolipid are non-saccharide, amino sugar and stearic acid. Fatty acid present in glycolipid are mostly celebronic acid, nervonic acid, ligoceric acid, etc. They do not contain glycerol and H3PO4. Sphygmol fatty acid are carbohydrate. Depending on the nature of carbohydrate component, glycolipids can be classified into two parts. Cerebrosides, here simple carbohydrates monosaccharide, represents the carbohydrate component. For example, glucocerebroside, galactocerebroside. Gangliosides, here complex carbohydrate, amino sugar, sialic acid along with monosaccharide presents the carbohydrate component. Sulfolipid, the sulfolipid or sulfoglycolipid. These are glycolipids with SO4 group. For example, sulfocerebroside, glucocerebroside CO4, galactocerebroside SO4. Lipoprotein, they are composed of lipid and protein called apoprotein. Lipids present in lipoprotein are TAG, triglycerol, cholesterol ester, CE, free cholesterol and phospholipid. Important lipoprotein are chylomicron, largest and least dense lipoprotein, VLDL, very low density lipoprotein, LDL, low density lipoprotein, HDL, high density lipoprotein, smallest and most dense lipoprotein. Important lipids present in neural tissues are lecithin, caphalin, saphenogomyelin, cerebroside, sulfobroside, ganglioside. Neutral lipids carry no charge at normal body pH. For example, TAG, cholesterol ester, monoglyceride, and diglyceride. Sphygmolipid definition: it is ceramide fatty acid plus sphygmol based complex lipid. Types: sphygmomyelin, ceramide, H3PO4 choline. It is phospholipid. 2 glycolipid. Ceramide carbohydrate. It is not phospholipid. Important function: component of biological membrane. Sphingomyelin constitute myelin sheath. Glycerolipid, phospholipids. Sphygmolipid definition: it is ceramide fatty acid plus sphygmol based complex lipid. Types: sphygmomyelin, ceramide, H3PO4 plus choline. H is phospholipid. Important functions: components of biological membrane. Sphingomyelin constitute myelin sheath. Glycerolipid. Phospholipids is sphingolipids. It is glycero-based lipids, H3PO4 based lipids, ceramide based lipids. For example, TAG. For example, glycerophospholipids. For example, sphingomyelin. Classification of lipid based on polarity. Non-polar lipid. They are hydrophobic and water insoluble. No part of this group of lipid shows hydrophilic character. For example, TAG. Polar lipid. In general, lipids are water soluble since they contain a predominant non-polar hydrophobic hydrocarbon group. However, some lipids along with their non-polar group contain some Non-polar group contains some polar hydrophilic group. Therefore, part of their molecule is hydrophobic, water insoluble, and part of it is hydrophobic, water soluble. Amphithetic polar lipids are the lipids having both polar hydrophilic and non-polar hydrophobic groups and shows limited degree of water insolubility due to their hydrophilic polar group. For example, fatty acid COOH group, phospholipid phosphate nitrogen base, glycolipid carbohydrate group, bile salt pH, taurine glycine cholesterol pH group, 2-MeG OH group, bile acid OH group. Fatty acids Fa. Fatty acid is carboxyl COOH group containing aliphatic acid. They are weak and with pH value of 3 to 5. They are acid because they contain acidic group COOH, which at pH 7.4 can ionize to release proton in solution and turns into carboxylate ion COO. Usually they are monocarboxylic acid having a long hydrocarbon chain with one term carboxyl group. The carboxyl group. They are acid because they contain acid group COOH, which at body pH 7.4 can ionize to release proton in solution and turns into carboxyl ion. Usually they are monocarboxylic acid having a long hydrocarbon chain with one terminal carboxyl group. The carboxyl group COOH is hydrophilic, but hydrocarbon chain is hydrophobic. So fatty acid shows amphipathic character having both hydrophilic COOH and hydrophobic hydrocarbon chain part. Usually they are monocarboxylic acid having a long hydrocarbon chain with one carboxylic group. The carboxylic group is hydrophilic but hydrocarbon chain is hydrophobic. So fatty acid shows amphitic character having both hydrophilic and hydrophobic part. Short chain fatty acid is water soluble because their hydrocarbon chain is short. For long chain fatty acid, the hydrophobic hydrocarbon protein is long, so they become highly water insoluble, because of which they are transported in circulation with plasma albumin. Of the total plasma fatty acid, 45% found on TIG, 35% in phospholipids. Fatty acid Fa. Short chain acid is water soluble because their hydrophobic hydrocarbon chain is short. For long chain fatty acid, the hydrocarbon protein is long, so they become highly water insoluble, because of which they are transported in circulation with plasma albumin. Of the total plasma fatty acid, 45% found on DAG, 35% with phospholipids, 15% with cholesterol ester, and 5% exist as free fatty acid FFA. More than 90% of fatty acid is in human body contains even number of carbon atoms, mostly with carbon number 14 to 24. Less than 5% of fatty acid is in human body contains odd number of carbon atoms. Source of fatty acid: endogenous source, synthesis of non-essential fatty acids within the body. Exogenous dietary source, for structure of fatty acid: animal fat, milk, butter, ghee, dalda, coconut oil, palm oil, banaspati. Hydrogenated fat, for unsaturated fatty acid: vegetable oil, fish oil, cod liver oil, egg yolk. Classification of fatty acid: A. Based on total number of carbon chain length. A short chain fatty acid carbon number 10 or less, mostly found on milk, for example acetic acid, propionic acid, butyric acid, etc. Long chain fatty acid carbon number 10 or more, for example palmitic acid 16C, stearic acid 18C, etc. Based on saturation of carbon. Structured fatty acid SFA. They do not contain any double bond in their hydrocarbon chain. They represent about 50% of body fat acid pool and usually remain solid at room temperature. For example, palmitic acid 16C, stearic acid 18C, butyric acid 4C, acetic acid 2C, unsaturated fatty acid UFA. They contain one or more double bond in their hydrocarbon chain. Double bonds are nearly always in cis form to long chain fatty acid carbon number 10 or more. Palmitic acid 16C, stearic acid 18C, etc. based on saturation of carbon. Saturated fatty acid SFA. They do not contain any double bond in their hydrocarbon chain. They represent about 50% of body fatty acid pool and usually remain solid at room temperature. For example, palmitic acid 16 degrees Celsius, stearic acid 18 degrees Celsius, butyric acid 4 degrees Celsius, and acetic acid 2 degrees Celsius. Unsaturated fatty acid. They contain one or more double bond in their hydrocarbon chain. Double bonds are nearly always in cis form, not in trans form. Page 22 and 75. In natural occurring unsaturated fatty acid, they represent about 50% of body fatty acid pool and usually remain liquid at room temperature. Unsaturated fatty acids are classified as mono monosaturated fatty acid MUFA. For example, aloic acid 18C, palmitic acid 16C. They contain one double bond found in olive oil, canola oil. Polysaturated fatty acid PUFA. For example, essential fatty acid. They contain two or more double bond found in fish oil and vegetable oil except coconut oil and palm oil. Nutritional classification. Non-essential fatty acid. They are the fatty acids which human body can synthesize. For example, palmitic acid, stearic acid, oleic acid, etc. Essential fatty acid. They are polysaturated fatty acid PUFA which are not produced in human body or must be supplied with diet. It is wrong to say that they are essential fatty acid because they are essential for body. In fact, all fatty acids are essential for body metabolic point of view, but essential fatty acids are regarded as essential because their presence in diet is essential since they are not produced in body. Essential acids for human body are linoleic acid, it is 18 degrees Celsius, omega 6 fatty acid with two double bonds. Linoleic acid, it is 18 degrees Celsius, omega 3 fatty acid with three double bonds. Arachidonic acid, it is 20 degrees Celsius with omega 6 fatty acid with 4 double bonds. Common sources of essential fatty acid. Vegetable oil except coconut oil and palm oil. For example, soybean oil, mustard oil, etc. Fish oil, fish viscera. It is rich on omega 3 fatty acid, inulinic acid. Cod liver oil and egg yolk. Source of linoleic acid. Biological importance of essential fatty acid PUFA, omega 3 and omega 6 fatty acid. Acts as precursor of icosanoids, for example, prostaglandins. Acts as components of biological membrane and maintains its fluidity. In human body, group 2 icosanoids are mostly prevalent. Reduces plasma cholesterol by increasing cholesterol excretion via bile by enhancing cholesterol oxidation to bile acid. Two essential fatty acids are antitherogenic and reduces the risk of coronary artery disease. Helps in gonadal functions and reproduction. Helps in vision and growth.